We live in a world that sometimes seems out to get us, even on the chemical level. The United States Center for Disease Control estimates that as many as 1 in 4 Americans are poisoned by their own food each year, though, of course, most of them recover. Now, bacterial infection can release toxins into your systems as well, and that can make us ill or even kill us. And just think of how terribly afraid we can be of animals a fraction of our size because they possess venoms meant to immobilize prey, but so powerful they can kill us if the animal strikes in self-defense. You probably noticed that I touched on all 3 words from this lecture's title just then: poisons, toxins, and venoms. All chemical compounds or cocktails, all naturally occurring, and all potentially deadly. In casual conversation, we frequently interchange these terms. But the truth about these 3 classes of chemicals is that they are not interchangeable. There is a hierarchy of classes, each defining a subset of the last. Poisons can be thought of as any chemical that disrupts the normal biological functions of a living system. Classics in this category are things like arsenic and lead. Toxins represent a subclass of poisons. Toxins disrupt biological systems, but are created by another biological system. Probably one of the most well-known human toxins is that from the botulinum bacteria. It's a compound that causes paralysis and inflammation in humans. Now, even organisms as simple as the castor oil plant can create toxins. The seeds of castor oil plants are the source of the well-known toxin ricin. Our third class of compounds for our discussion today is venom. And a venom is most simply defined as a toxin that one animal transfers to another by injection, either as a predatory or defensive tactic. Classic examples of venom are the potent chemical cocktails used by spiders, scorpions, and snakes to immobilize prey or attackers. All 3 classes of chemicals from this hierarchy, poisons, toxins, and venoms, are worth understanding, and not only so that we can try to prevent or treat their ill effects. As we'll soon see, centuries of study have researchers poised at the brink of developing technologies that can not only defeat these poisons, but put them to use for the benefit of mankind. So let's spend this lecture getting familiar with some of nature's most potent poisons, toxins, and venoms. In 1538, a man by the name of Paracelsus, a Swiss physician and the first recognized toxicologist in human history, famously wrote, "The dose alone makes the poison." He makes an interesting point. Every meal that you eat contains trace amounts of arsenic, yet you don't seem to suffer any ill effects. But consume a pea-sized grain of this infamous material, and it's off to the emergency room for you, not the morgue. Let's look at the other extreme. Although it's rare, several times each year, people actually die from heavy overconsumption of water. Usually, the victim of a poorly researched radio contest or fraternity hazing ritual. Yet, on a hot day, most of us gulp down water without ever thinking twice. Now, these examples prove Paracelsus' comments to be true when taken to extremes, but our colloquial definition of poison is a bit more even-handed. Simply put, poisons are substances that have negative health effects when we're exposed to them in concentrations or amounts that are commonly encountered. Paracelsus' unusual definition aside, some things about poisons are incontrovertible. They have played a profound role in the progress of human history, perhaps most famously as a tool of assassins throughout the histories of the Greek, Roman, and Byzantine empires. Poisons like the hemlock plant and venom from snakes were used as instruments of suicide by prominent historical personalities like Socrates and Cleopatra. In fact, it might seem that the only thing that influential peoples of ancient Greek and Roman empires did better than disagreeing with one another was to settle their scores with these silent killers, poisons. Poisons continue to influence our behavior as a species even today. Although the rather nefarious practice of political assassination is far less common in the modern world, it does still happen from time to time. Bulgarian writer and political dissident Georgi Markov was felled in 1978 by a small pellet of ricin, craftily injected beneath his skin by a mysterious, umbrella-carrying man in London one evening. Or the story of Alexander Litvinenko, a former Russian organized crime investigator whose defection to the West was cut short using just 10 micrograms of polonium-210, a highly radioactive isotope that killed him using radiation poisoning from the inside out. Most of us are familiar with classic poisons like arsenic, cyanide, and thallium. These are inorganic, non-biological materials so acutely toxic that only a small amount of them is needed to bring about disastrous health effects. And as interesting as other poisons like ricin and polonium may be, any lecture on poisons simply has to start with the granddaddy of them all, arsenic. There's evidence that in the 1st century AD, arsenic in its sulfide form had been discovered and used by Greek physicians to aid in tooth extraction. But it was quickly recognized that ingestion of large quantities of arsenic-containing compounds had catastrophic effects on human health. It led to violent abdominal cramping, vomiting, and often death from the shock that resulted. It was Paracelsus himself who published the first detailed instructions for the production of metallic arsenic from its naturally occurring sulfides, and this launched it to the forefront of poison technology. Thanks to his work, arsenic quickly became the poison of choice for anyone with the resources to get it and a score to settle. The potency of poisons is characterized by their LD50. This is the lethal dose for 50% of a population. Arsenic's LD50 of about 15 milligrams per kilogram in rats translates to about a 1 gram dose being potentially fatal for a human of my size. That is a grain of arsenic about the size of a jelly bean. A more perfect poison for its day couldn't be wished for. Arsenic remained the poison of choice in Western society for many centuries. As recently as the Renaissance, this colorless, odorless, tasteless material was used so frequently that it gained the nickname inheritance powder. So, how does arsenic do its damage? What is it about this element in particular that makes it so dangerous when consumed? It's a pretty simple material. It's an element itself. So how could it have such a potent, focused, and specific biological effect on us? The answer lies just above it in the elemental group on the periodic table. There we find phosphorus, an element that often appears in biochemistry as phosphate. Now, phosphates serve multiple critical roles in biology, including holding your DNA together, as well as playing roles in the storage and use of energy. It's that last function that makes you and me vulnerable to arsenic. You may have heard of adenosine triphosphate, or ATP, a compound that helps your body to manage energy storage. Now, ATP is an adenosine base with 3 phosphate groups attached to it in a chain. Removal of a phosphate group creates ADP and a considerable amount of chemical energy that your body taps to keep itself going. Now look at this complex ion, arsenate. Look familiar? Of course, because arsenic has a 4s2, 4p3 electron configuration, which makes it very similar to phosphate. So similar that it can replace phosphate in ATP molecules, causing a malfunction in the energy storage and transmission process in your body. The next stop on our tour of the less pleasant side of modifying our body's chemistry is toxins. Remember that toxins are a type of poison, but one that is generated biologically. Recall that arsenic, possibly the most famous poison in the world, is taken from an inorganic mineral source. It's simple, elemental even, in its composition, but it wreaks lethal effects on a wide array of systems in your body. But life has had a lot of practice at creating newer and deadlier materials, often for the purpose of self-defense or the hunting of prey. In the nearly 3 billion years since life formed on this planet, a battle has ensued, one that pits living things against each other in the struggle for existence. certain plants and animals have been able to hedge their evolutionary bets by creating substances that make them less attractive as a food source for others, or even that go on offense, beating back competing life forms to increase the chances that they themselves will have enough resources to survive. A classic example of toxins are those compounds created by plants like hemlock. Hemlock plant produces copious amounts of this molecule, coneine. Coneine acts like many drugs, binding to proteins in your body that need to function properly to keep you alive. It actually impersonates nicotine, another toxic substance found in tobacco plants. Now, both of these molecules bind to receptors that process ions in your body. Usually, the cause of death is respiratory paralysis, as the muscles that flex to fill and empty the lungs fall still. Nicotine itself is produced by tobacco plants to discourage insects from eating them. It's a natural defense mechanism for the plant. And we tend to think of nicotine as a relatively weak toxin compared to others, but in fact, the proper administration of just a small amount, not much more than you would find in a cigar, can be fatal. So fatal, in fact, that poisoning by nicotine has occasionally been the topic of high-profile murders. In 1994, the wife of a nuclear power plant worker, whose name was Paul Curry, passed away very suddenly, leaving him with a substantial life insurance payout as a result. But investigators discovered a dubious plot to end his wife's life using a substance so common and apparently harmless that it would be overlooked. Mr. Curry's weapon was nicotine. With an LD50 of about 1 milligram per kilogram, it takes just about 60 milligrams of nicotine to take down the average human. That is the amount in just about 3 typical cigarettes. Of course, smoking that amount of tobacco only transfers a small fraction of that nicotine into a person's bloodstream, but an injection is far, far more efficient. The dose makes the poison indeed. And plants aren't the only members of the biosphere that have adopted this kind of chemical survival strategy. Animals have their share of potent toxins as well. In fact, the most deadly toxin yet isolated came from the back of one of these little critters. Here it is, batrachotoxin, excreted by the glands of the poison dart frog. This compound acts by binding to proteins that transport sodium ions across cell membranes as part of normal muscle contraction. The mechanism of action is actually quite common among toxins and poisons, but what makes batrachotoxin so special is its potency. This molecule binds so powerfully to sodium ion channel proteins with a KD of about 10 nanomolar that the LD50 for it is estimated to be about 2 micrograms per kilogram. That means that someone my size could be killed by far less than a milligram of this toxin. A mass 1,000th the size of a pea is really all it takes. That is remarkable potency. This toxin has garnered more than a bit of fame for its use as a tool in hunting by the indigenous peoples of Central and South America. Just a drop of the poison secretion on an arrow should be enough to take down even the largest prey in one shot, once an arrow delivers it to their bloodstream. Because the toxins produced by these plants and animals are relatively simple small molecules, they tend to be capable of surviving a trip through the GI tract, making them truly poisonous. All a predator has to do is take a taste of one of these little creatures, and their life is in very real jeopardy. The sting of a scorpion or the feared box jellyfish, the bite of a copperhead snake. Not the kind of experience that any of us would sign up for, but a harsh reality for those of us unfortunate enough to tread in just the wrong place at just the wrong time. Just one sting or bite from any of these animals can take the life of an animal hundreds of times their size. It's a fearsome thought, one that's the stuff of legend, from Hollywood horror movies to cautionary urban tales. It seems that many of nature's predators have developed systems to make their bite or sting as lethal as possible, not by the direct damage done by that bite or sting, but by potent chemicals introduced at the site of the wound. It's quite remarkable, isn't it, that animals like these can produce and carry around a venom so toxic that it can kill, yet they themselves are unharmed. The venom kills prey, which the predator now eats, and the predator is just fine. It's intriguing, isn't it? But how does it all work? Well, as usual, nature is way ahead of man on this one. Venoms from predatory animals are not only very potent, they're far more complex at the molecular level than most poisons are. In short, venoms are proteins. They're made of the same amino acids as the rest of the proteins in our bodies. Now, once their job is done and the predator starts to chow down, the predator's digestive tract treats that venom like any other protein, breaking it down into amino acids for nutrition, and this renders it non-lethal. But before all that happens, the venom is injected into the prey, bypassing the digestive tract altogether and surviving the transfer intact to paralyze the muscles of the prey, in some cases leaving it defenseless to further attack, and in other cases, killing it outright by stopping its heart or lungs from functioning. So, how does the venom accomplish this? Well, the answer to that question is going to require that we first consider some of the chemistry behind muscle contraction. See, when your muscles flex, be they voluntary like your arms and legs or involuntary like your heart, the cells making up those muscles are responding to the flow of certain ions across their cell membranes. But that raises an issue. Cell membranes are made primarily of molecules called phospholipids. These phospholipids come together to form a very low polarity barrier that holds the contents of the cell inside. Now consider this. How do you get a charged potassium ion, for example, to cross that barrier? On the outside of the cell, the ion's happy, solvated and stable, interacting well with the dipoles of the water molecules around it in a favorable ion-dipole interaction. But for muscles to function properly, that potassium has to cross the membrane, sacrificing all of that stabilizing interaction with solvent, only to be surrounded by long hydrocarbon tails that do not have any polar bonds to speak of. It's an environment that potassium ions simply aren't going to become a part of, not even for a split second as they move into the cell. But cells have developed a clever way to solve the problem. Cells use special proteins called ion channels. Now recall that some amino acids have very low polarity side chains and others very high polarity side chains. When inserted into a cell membrane, a folded ion channel protein presents a very low polarity face to the hydrocarbon tails of the phospholipids, allowing it to sort of embed itself into the cell membrane. Now at the top and bottom are higher polarity side chains interacting with the water on either side of the cell membrane. And most importantly of all, the interior of the channel itself is loaded up with polar bonds just waiting to interact with potassium ions favorably as they make their trip across the membrane. Problem solved. An example of this is the potassium channel shown here, solved by the lab of Roderick McKinnon in 1999. Notice the leucines, valines, and other low polarity side chains adorning the outside to help it rest comfortably in the cell membrane. Now, when in place, this magnificent structure can transport potassium ions across the cell membrane to help keep your muscles working correctly. But the powerful short proteins or peptides in the venom of scorpions shut these remarkable creations down with deadly efficiency. The venom of these and other animals contains short peptides that are capable of interacting with those ion channels. One example of this is the peptide known as chlorotoxin. Now, being peptides or short polyamides themselves, many of the active compounds in these venoms can carry out very focused chemistry just like their larger protein brethren. In the case of scorpion venom, the potassium channel is the target. The chlorotoxin seeks out and binds to the opening to a victim's ion channel proteins through a set of very specific side chain interactions between those two molecules. Acting not unlike a drain plug, chlorotoxin caps off ion channels and cuts off the ion superhighway that the cell uses to manage energy. And no energy means no muscle contraction. With the peptides from the venom in place and your muscles no longer functioning, you're at a serious risk of death because your lungs can no longer fill with air and your heart can no longer beat properly. The protein from scorpion venom that does this is relatively small, but it is a protein, and it has a very specific fold that gives it its activity. In fact, most animal venoms are protein-based. Because they're meant to be injected into a victim's bloodstream by a bite or sting, this chemical strategy will work. The venom bypasses the digestive system, goes directly to work, paralyzing victims by blocking the flow of ions across their cell membranes. Now, as bad a reputation as venoms have, they also have incredible potential for positive applications. In particular, these molecular assassins are so potent and lethal to human cells that researchers want to find ways to harness that lethality to kill cancer. You see, cancer cells often grow faster than ordinary cells, and they have a greater need for resources. The increased need gives some cancers a tendency to overproduce certain proteins that regulate their energy. And proteins like potassium channels fall into that category. Now, what does this mean for cancer cells when chlorotoxin is in play? Well, it means that they may be more susceptible to its deadly effects. Chlorotoxin might just be used as a medicine to treat certain kinds of cancers. But this leads researchers to a new problem. With something as deadly as chlorotoxin, delivery to a tumor has to be as direct as possible. Allowing this deadly molecule to wander aimlessly in the bloodstream of a patient is asking for trouble. So researchers have begun working on ways to attach chlorotoxin to specially created delivery vehicles called nanoparticles. Made of iron oxide and just a few nanometers across, these special particles have a remarkable tendency to accumulate in cancer cells, preferentially when introduced to a patient's body. Now, combine that with the fact that they can be chemically tethered to a handful of chlorotoxin molecules, and you have a potential strategy for targeted delivery of chlorotoxin to cancer cells. As the nanoparticles accumulate around tumors, their payload of scorpion venom does its work on the overexpressed potassium channels on the cancer cells. In theory, increasing the efficacy of the toxin on the cancer while minimizing its effect on healthy cells. It's a brilliant strategy, but it's a new one. Research continues to see if this marriage of one of mankind's most recent creations, the nanoparticle, and one of nature's oldest, venom, might create life-saving cancer drugs someday. So let's review our discussion. We started thinking about the power of the chemical compounds and even elements have to affect our health. Not positively, as is the case with medicine, but negatively. We discussed how we define any material that's bad for our health as a poison, creating a very broad class of materials. We took a look at one of the all-time classic poisons, arsenic. And we saw how just a small dose of this element can be deadly because it impersonates phosphorus in our systems, interfering with chemical pathways that are used to transmit and manage energy. Then we took a look at a narrow class of compounds, toxins. Toxins are poisonous materials created by other living systems to give them an evolutionary advantage. We looked at chemical cousins codeine and nicotine from hemlock and tobacco. And we took a look at the structure and action of batrachotoxin, the most potent toxin known to man. Then we turned our attention to venoms, poisons that are only effective when injected directly into the bloodstream of the intended victim. Usually this happens through a sting or a bite. And we saw that venoms get their remarkable potency from the fact that they are themselves proteins, complex biopolymers slowly perfected for their purpose through eons of evolution. But we also saw the weakness in this strategy, that venoms are typically less poisonous when ingested. Finally, we blurred the lines between medicine and poison, and we took a look at an example of how modern scientists and physicians are finding new ways to apply nature's most deadly chemical creations in a quest to cure cancer. So today we saw how bacteria, plants, and animals have devised ways to wage chemical war on one another as they compete for biospace and food. The natural progression forward is clear. Next time, we're going to turn our attention away from nature's creations and toward those of man. We'll explore a dark side of human pursuit of science of chemistry as we investigate man-made chemicals as weapons of war. Well, now that we've completed classifying poisons, toxins, and venoms, let's think a little bit more about that hierarchy and do this in terms of a challenge problem. So here's our challenge problem. Let's classify a few different substances and creatures as poisonous, toxic, or venomous. Now remember that poisons are anything that are bad for health, that toxins have to be biologically created, and venoms actually have to be injected and cannot be ingested or eaten. In other words, we can create a Venn diagram, if you will, for these kinds of materials in which we have venoms in the smallest region, toxins next, and finally we have poisons. So all venoms fit within the poison circle, right? All toxins fit within the poison circle, but not all poisons fit within the toxin and venom circles. So let's think about some of these different animals and materials in terms of whether they're poisonous, toxic, or venomous. Now, in the case of things like mercury and lead, of course, these are non-biological materials. And so lead and mercury would fall under strictly being called poisons. You would never call lead or mercury a toxin or a venom to be absolutely accurate. They form without the assistance of an organism. How about some toxins? Let's see if we can pick a few toxins out of here. Now, here's an interesting one. Monarch butterflies can actually be toxic because of steroidal compounds that they ingest in the larva stage that stay with them through life. So, for certain animals, consuming a monarch butterfly can cause them to become very, very ill. Similarly, pufferfish are a source of a compound known as tetrodotoxin, which is, of course, a toxin. It's very bad for you if you ingest it. So, pufferfish and monarch butterflies themselves could be considered toxic because if you eat them, you will have some deleterious health effects. And that leaves 4 more on our list: black widow spiders, vipers, box jellyfish, and scorpions. So why have I left them off until now? Well, I've left them off until now because, of course, these particular animals have to inject their toxin into an organism in order to make it work. So while you could say that a black widow or a viper produce a toxin, it would be more accurate to say they are venomous. Again, we have to call them venomous because, yes, they do fall into a poisonous portion of the diagram. Yes, you can say that they create toxins because they're animals that are making compounds that are bad for our health. But to be truly rigorous about classifying them, we would call them venomous because they belong in this smaller part of my diagram here, in which we have a biologically created toxin that must be injected into another organism in order to take its effects.